You're listening to Let's Talk Creation with Todd Wood and Paul Garner, the creation show where we learn, grow, and worship.
Well, welcome everybody to another episode of Let's Talk Creation with Paul Garner and Todd Wood. I'm Paul Garner.
And I'm Todd Wood.
It's great to be back again with you. Do remember to like our episodes, share them with your friends, subscribe, hit that subscribe button if you haven't already. If you're a new listener or viewer, you're especially welcome. And we say hello once again to all our regular listeners. It's great to see the audience growing, and we can only do that with your help. So please do share the word about the podcast and tell other people about us. That's really great. Today, we are going to be interviewing another very special guest. We're going to be thinking about a topic. I guess this could be part of our Great Discoveries in Creationism series, Todd, that
I was
we pick
just
up every
thinking
now
that.
and then.
Yeah, I was just thinking that this is definitely a great discovery in creationism.
Yes, absolutely. So, yeah, we're going to be thinking about some research that's been going on, and it has some really interesting implications for how we think about geological timescales. And so I hope you'll really enjoy this episode as much as I'm sure we're going to have fun recording it. Our guest today is Professor Leonard Brand. Leonard is Professor Emeritus of Earth and Biological Sciences at Loma Linda University. And Leonard, this is your first time on the podcast, but it's really great to have you. We're really glad that you could join us today. Welcome.
Yeah, well, I'm glad to be here.
So let me just say, we've been talking
Sure.
about having you on the podcast basically since we started.
Yep.
And here we are 120 some episodes in, and we finally got around to it. So I guess there was a lot more to talk about creation than I anticipated when we began, but better
Yep.
late than never, I suppose.
Yeah, absolutely. Yeah, no, we've wanted to have you on the podcast for a very long time. And Leonard, for those of our listeners who may not be familiar with you and your research, just tell us something about yourself and the work that you've done, your career, because it's a fascinating sort of long career. You're well published in some really interesting disciplines. Just tell our listeners a bit about that.
Well, I got a PhD in evolutionary biology from Cornell University, and then began studying chipmunks. And a few years later, Harold Coffin gave me a handful of papers on the Coconina sandstone and asked me to read them, and on the fossil tracks there. And I realized there's research that needs to be done, has been done. And this actually made it a rather big change in my career. I retrained in geology and been studying fossils and rocks ever since. And this is one of the projects that we've done that we're going to talk about today.
Yes, and of course, we're not going to be talking about the Coconino, but that's one project that a lot of people will know that you've done research on the vertebrate trackways in the Coconino, particularly. That's been a particular sort of focus of your research, but there are so many other really interesting sort of research areas that you've worked in, and yeah, at some point we're going to have to talk about some of those as well. But today we wanted to have you on the podcast to talk about something called biotubation. Now, I can already, our viewers and listeners, may not be familiar with these technical terms. And before sort of people start thinking, I don't know what biotubation is, let's begin there. What is biotubation? Because this is something that is actually really significant when it comes to thinking about geological timescales. And so this is really exciting work. So tell us, what is biotubation?
Okay, well, I'll start with last fall. I spent a morning sitting on a beach in Hawaii watching little crabs scurrying around on the beach. And a wave would come up and it would destroy their burrows. And they immediately make new burrows and then go about their activity. So they're burrowing into the sand. And then there are a host of little creatures in the sand, on the sand, burrowing around, walking on the sand, burrowing. Um, and eventually they will destroy any structures that are in that sand. That's bioturbation, the activity of animals working through the sand and eventually kind of homogenizing it. And so we studying what they're doing and how long it takes is what we're about.
Okay. So what kind of animals are we talking about? What animals biotobate sediment?
Well, little crabs, types of marine worms, quite a wide variety of small creatures that live in the sand or are partly on the sand, and they live there and they get their food by burrowing through the sand and picking up particles of food.
So these animals are kind of burrowing for food, and presumably some of them construct places to live, the dwelling burrows as well. Is that right?
Yeah, dwelling burrows, feeding burrows, escape burrows, a wide variety of things. It's actually a huge topic. I mean, there's a massive literature on it. But I just summarize a few things.
Yeah.
Yeah.
And
So,
Yeah.
also, I'm talking about marine environments. You have the same thing on terrestrial environments. The ground
mm,
is continually burrowed and changed by pocket gophers and mice and squirrels and a myriad of worms. They're doing the same thing. But most of the study on this is with marine situations, aquatic environment.
so do footprints count as bioturbation? Trackways?
Yeah, they do.
They
They
do?
do. I don't. Yeah. So it's mostly dealing with invertebrates or what they do in the
Okay.
sand and
Okay.
I have heard the expression dinotubation, which is kind of where dinosaurs trample surfaces and
Wow.
leave footprints, and so they
yeah.
can kind of churn things up too. But
Yeah,
I
so I was saying the literature is mostly on invertebrates, but it's definitely vertebrates do it, and uh, dinosaur tracks churning up the surface is a dinoturbation.
yeah, yeah. One question I wanted to ask you is, these organisms when they burrow into the sediment. I mean, how deep down do they burrow? How deep are some of these burrows in the sediment?
Oh dear, that's there go centimeters, a meter perhaps. That usually not more than that.
Wow.
Hmm.
They're working through the top part of the sediment.
Yeah. So another sort of expression that people may have heard is this distinction between body fossils and trace fossils. Um, just walk us through that. What's the difference between body fossils and trace fossils, and how does this relate to biotubation?
Okay, body fossils are the fossils of the organism. Their skeleton, any part of them that gets preserved. Trace fossils, or sometimes called ichnofossils, they're results of activity of the animal, marks that they leave. It's not part of the animal itself. It's something they do that gets preserved. Tracks, burrows. I guess coprolites are in that category, but they're a little different type of thing. So
Yeah.
they're results of the activity of the animals. They're not the body itself.
So biotubation is in effect, I guess, caused by an accumulation of trace fossils in sediment. That's what biotubates a sedimentary rock, right?
That's right. That's what it is.
Yeah.
Trace fossil is what we call those features.
Mhm.
Uh, the result of all of that is bioturbation,
Yeah.
churning up sediment, changing the sediment.
Yeah, so for our listeners, what we're talking about here are these marine animals and also terrestrial animals too. They'll do it too, but they colonize freshly deposited layers of sediment and they will burrow into it, and this churns the sediment up, so it sort of destroys any structure, any layering, any sedimentary features in those layers. How effective are they at doing this? I mean, how quickly do these animals get to work? Imagine there's if we imagine there's a sediment layer deposited on the floor of the shallow ocean, maybe there was a hurricane or something deposited a layer of sediment. How quickly does that sediment get churned up by these things?
That's a very good question, and then the answer is interesting. Uh, and there's a lot of work been done on this, and it depends on how abundant the bioturbators are. But
Mhm.
the bioturbation, the churning of the sediment, um, and until it actually destroys any structures in there, it takes hours or days or weeks or maybe months, not years. It does not take that.
Mhm.
It happens quite fast.
And I mean, are we talking about large numbers of animals doing this? I mean, what's the kind of density of animals that you need to completely biotubate,
Uh,
a meter thickness of sediment, for example?
well, there's studies that report this, and it can be anywhere from say 15 to hundreds or thousands even in a square meter of sediment.
Wow.
Usually not that high, but there are quite a few of them in there.
Yeah. So they get to work really, really fast.
Yes.
And so sediments basically don't remain pristine for very long if they're exposed to, you know, this kind of colonization by invertebrates.
No, in normal environments, it doesn't take long before it's churned. And then these creatures don't only go into new sediment, but they live there.
Mhm.
And so the ocean is working all the time, and new particles of food get put in. So they're continually working and finding food in there and making new biodegradable.
So
Now,
when you so can I
sorry. Go,
ask a question
sure,
here?
go ahead, Todd.
Yeah.
Yeah.
So when you say work through a sediment, so I mean I've seen pictures of like lake sediments where there's these tiny little layers that are supposed to be annual layers that go for meters of thickness. You're describing something that kind of homogenizes like a storm deposit in a marine environment. So we're not talking about lake sediment. We're talking about ocean sediments. And we're talking about, say, some big storm comes along and dumps some sand, and then these guys come in and bioturbate it. Are they destroying those layers? Is that what you would expect to see? That there's basically no layering after they're done?
Yeah, the much of the research, maybe the majority of the research is in marine environments, but it also happens in freshwater environments. And they're working through the sediment all the time. But when some new sediment comes, then you probably get layers deposited, and you can tell the boundaries between the layers. But when the bioturbators start working, they begin to chew through those boundaries until eventually you don't see any boundaries between layers. And that's the part that is especially important here for what we're talking about.
Mhm.
Um, you can see, you can get some idea of how long it takes because it works pretty fast in the marine environment.
Right. Okay.
And though when you talk about these persistent layers,
Hmm.
Mm.
that that means you have not had a lot of bioturbated working through there
Yeah.
for very long
Yeah.
because those layers would be gone.
Right. Yeah.
Yeah.
They'd have to be. Yeah.
Mhm.
Okay.
Now, as I've sort of read the anti-creationist literature, I have seen the argument that bioturbation is actually a problem for creationists, that it's a problem for flood geology, because we have bioturbated layers throughout the geological column, and these layers require some time to form. And so this is a challenge for flood geology. Just kind of help to unpack that. Why do people see bioturbation as a problem for the creationist viewpoint?
Well, I would say if they say it's a problem, they haven't really looked at it quantitatively.
Mhm.
Uh, you know, because like with the research that we'll talk about, um, these creatures destroy the sediment pretty fast, the layering sediment pretty fast. And when think about the flood, things are happening very fast. Okay, so the flood is tearing up sediments, and it's ripping up, taking out of place all these little organisms. Okay, what are they going to do? They're going to look. They're going to be in the water, moving around, and they'll look for a place to settle, a place to walk, a place to burrow. And if they can find it, they will start burrowing or doing something. And so it's actually not at all surprising that there's bioturbation all through the call. I would expect that. The question is how much, and how much bioturbation there is gives an indication of how much time there has been for things sitting quiet. And that amount of time is what we'll talk about in our main research that we'll report here.
Yeah. I guess people see the challenge in terms of how many bioturbated layers there are. I guess they're thinking in terms of there being hundreds, thousands of layers of bioturbated sediment, and this requires some time. So it's really interesting that you've done some quantitative work to try to put some numbers on this and actually sort of pin this argument down.
And I think it is not. It's not a problem that there's bioturbation all through the geology column, because this is happening all the time. Organisms are being ripped up and moved, and wherever they are starting from, they will look for a place to settle.
Yeah. Yeah.
This will happen all the way through the geological column.
Yeah. So if we see sediments in the record, which we do, where layering and other structures are well preserved, how could we explain that? I mean, what possible sort of options do we have to explain the absence of bioturbation in sedimentary rock layers?
Well, there are a couple of processes that could reduce the amount of bioturbation. One is if there's just no oxygen. That makes it hard for the creatures to be there. Now, there have been research showing that even with low oxygen, there still is bioturbation in modern sediment. But if there's no oxygen, that's a problem. The other thing is, if the sediment is being deposited too fast for the bioturbators to get in there and do their thing, then you can have little or no bioturbation.
So
What about
can we test
Sorry, go
for
ahead.
can we test sediments for an absence of oxygen? Is there a way to look at a sediment and go, that was formed with no oxygen? Is that possible?
Uh, People do make those conclusions. I'm not familiar with all the ways that they test that,
Back.
but they do reach decisions about how much oxygen there was in modern environments.
um, I mean, are there environments in the modern world where sediments are being deposited where you just don't have bioturbators for whatever reason? I mean, I'm thinking about maybe deep marine environments, very anoxic environments, where you do have a lack of oxygen. Do we have modern examples of this where we have sediments with basically no bioturbation going on because the animals simply aren't there?
Well, the idea of no bioturbation, I'm not sure about that. Um, Certainly, some sediments there are less than others, but I haven't studied the depth of the literature enough to really say whether there are none.
But in shallow marine environments, which is, where a lot of sediments in the record are thought to have been deposited, you basically have an abundance of animals there, I guess.
Yes, generally, unless it really is, these two conditions I talked about that could prevent it. But otherwise, the normal thing is that there's plenty of bioturbation.
Yeah. Yeah. So in the modern world, where, a sediment might be laid down by, tides, or it might be laid down by, some unusual event like a hurricane or whatever. Typically, there's then some kind of time gap between that sedimentary event and the next one. That might be a few years, it might be 100 years, might be more. What would happen to that sediment? Would we expect sediments to be basically completely churned up, completely homogenized? So you're really seeing no structure in those sediments at all?
That's what ultimately you expect. And it isn't only storms that move the sediment along the coast here. Sediment, the sand is continually being moved. The nature of the currents are moving it gradually, moving it. I think around here, I believe it's moving south. Okay, so this is a continual process that's happened, and bioturbators are working through it while this is happening.
Yeah, I remember reading one textbook on trace fossils. It was by Bromley, I think.
Yes.
And in that book, I think he said, we kind of expect 100% bioturbation of a sediment to be the norm in the modern world.
Yes, he said that very clearly. That is the norm. If it
Yeah.
isn't 100% bioturbator, you need some explanation as to why.
So that's interesting. So I guess the challenge then is we can kind of compare the predictions of two different models here. So if we've got the creationist model where a large proportion of the sedimentary rock record was deposited during the flood, or at least deposited very rapidly, we would expect bioturbation to be less intense on average. It would be much more, much less common, much less abundant. But in the conventional viewpoint, where sediments are deposited sporadically over long periods of time, there ought to be lots of time for bioturbation to happen. So that's kind of that's really what we're saying. We've got really a test, haven't we, of the two models?
Yes, we'd like we said earlier, we would expect some bioturbation, but in our model, or in a biblical model, we would not expect to have very much intense bioturbation. Maybe some intervals, but not a lot of it.
Mm-hmm.
So
Yeah,
that ultimately is a test of the model.
yeah. So that brings us then to the work that you and others have been doing on this question of bioturbation to try to put some numbers, try to quantify this issue. That's one of the important things that we have to do as scientists. We have to try to put numbers on things. And so tell us about the work that you've been doing, what you set out to do, how you did it, and what did you discover?
Okay. Um, my friend Dr. Art Chadwick and I worked together a lot, and we were doing geology research in Utah and surrounding areas. And one of the questions that I had in my mind was, can we quantify bioturbation in the rocks? And so we tried it and decided, is this a worthwhile project to do? And so we worked our way up through a rock formation. We'd find a place where the rock was accessible. You had cliffs, or cliffs at least, that were navigable, and we could measure our distance, do what we call measure a section. You're measuring the height as you go up, and you're documenting things as you go up. And we document bioturbation centimeter by centimeter as we work our way up through this rock. And what we found was very interesting, and it led us to definite conclusions. This needs to be done for a lot of formations. So any formation that we could find where it had a good exposure for most or all of the formation, we could work our way up through without danger of bodily harm. We would do that. And we compared ultimately 37 different rock formations from Cambrian to Eocene, uh, making these measurements, quantifying the amount of bioturbation.
So is this
Now,
is this the grand staircase where you're doing this?
it's in that region, yeah.
Okay. Yeah. So basically, for those that don't know, this starts like at the bottom of the Grand Canyon, and you work your way up all through into Utah. It's a
Yeah, it's actually
continuous deposit, basically, is the point.
right. It's actually it's an unusual situation in parts of Utah and surrounding areas where well, usually as you read looking through a geological region, you don't necessarily expect the rocks to get younger and younger as you follow a certain direction of movement, because it falls to all kinds of things. But the Colorado Plateau, as it's called, and the areas above it and below it are unique. There's been very little tectonic activity.
Mhm.
And so the Rand staircase, as you go north, you're going up section.
Yep.
Up to younger rocks. And so this is a very unique region um, for study of this type.
Yeah. Now, for our viewers and listeners who may not be sort of quite so familiar with the geological column, you mentioned there that you work through rock formations representing the Cambrian to the Eocene. So in conventional terms, this is from about 540 million years ago to maybe, 55 million years ago, something like that,
Yeah, or younger.
or younger. So,
30 some, I don't remember exactly.
oh, okay. So you've worked through quite a good representative sort of section of the geological column.
That's right.
And it sounds like you you worked in some quite interesting places. So you're looking at vertical um, cliffs of rock. How did you manage to access all of those rocks to see vertical faces? Because that could be quite a challenge, I imagine.
You need vertical surfaces, but you find places where, for instance, it's not all vertical.
Hmm.
You can climb up, like you can climb up one surface going this way, but beside you are the vertical exposed surfaces. So you find
Okay.
things like that that make it accessible.
Yeah.
You
Yeah.
avoid rattlesnakes as far as you can.
Yeah. And I mean, this sounds like a really laborious task, because you said you were going kind of centimeter by centimeter, layer by layer through these
Right.
sequences. How long did this take you?
Oh dear. It isn't. It doesn't take as long as you might think. We maybe do a section in a day or a half a day, and then we go on to another place. And so
Okay.
it's intense work, but you can move
Hmm.
along.
Hmm.
Wow.
Yeah.
So how
Yeah.
much do you know offhand how thick these layers ended up being after you measured them all?
Yeah, actually, I made him add that all up, and it's over 5,000 meters of vertical distance that we cover, adding them
Wow.
all together.
Wow.
Yeah, 3 miles
Wow.
Yeah.
on doing.
Wow.
Yeah.
Okay.
Well, I
That's
mean,
impressive.
that's
It,
impressive. Yeah.
it,
Uh,
we spent a number of years doing this, so it
Yeah.
takes a while.
Yeah, yeah.
okay. And how many people were working on this? Were you doing it alone? Were you doing it with others besides Art Chadwick? How many were doing
Well,
it?
we had various students who were with us, and a couple of other colleagues that worked with us. So we usually had more than the two of us,
Hmm.
3 or 4 or 5.
Yeah.
Wow. Still a lot of work.
Yeah. It's a lot of work. It's a lot of work. So how did what you're trying to do is quantify the bioturbation. I mean, how does that work? Uh, is there some kind of standard scale for measuring intensity of bioturbation, or did you have
Yeah,
to invent one?
there is a scale that is used. There's about 4 or 5 different levels of bioturbation. We took that standard scale and modified it to fit our research approach. Because we were not, well, for one thing, we were not studying horizontal surfaces. We were looking at the vertical surfaces, and our question is there enough bioturbation to disturb the layering in the sediment? And so we had a scale of 4. First one, there's no bioturbation or a few burrows, but not enough to damage seriously the layer. And then second level 2, there's more, and 4 there's more, 3 there's 4 more, and then level 4 it's completely bioturbated. It's destroyed all the layer and everything there. And so we'd categorize that into those 4 different levels.
Hmm.
And of course, part of it we have to keep track of another part, and that is sometimes there is talent, slope, or other things that cover it, and you can't tell anything there. So we'd have to keep track of that as we went along as well.
Hmm. Hmm. And so you basically categorize the level of bioturbation in every bed within a formation. Is that kind of how you did it?
Yeah, every bit. We'd with a certain area that's exposed. We'd
Hmm.
measure how thick this is, and then we'd plot how what kind of bioturbation we find up through there. And if it was and we keep track of that. How much distance, how much vertical distance, or what was the bioturbation level?
Hmm. Hmm. And what did you find? I mean, what were the results of all of this work? Have you completed it, first of all? I mean, is this a project that's now finished, or are you still going out and measuring sections? Is this
Um,
ongoing?
we're we're finished with what we're doing. Now
Okay.
these projects are never finished. There's fascinating
Yes.
work that others could do to continue this, to ask new detailed questions. Yeah, but
Yeah.
we finished what we're going to do.
Yeah. Yeah. Okay, so what did you find? I mean, from your studies of this kind, what could you conclude?
Um, Actually, is the data were quite amazing to us that we ended up. So we have all this diagram, you know, each formation, what's there, what the distances were, and then we could determine the average for all of these. How much was in level one, what percent, and in all these others. And for 37 formations, the 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 amount of surface with our level one is 97%. So there's in level four we found I think less than 1%.
Oh my
So
goodness.
there were areas where we had an intense bioturbation, but not very
Hmm.
thick, and
Hmm.
so it's just very little. And this is amazing when you compare to what you read in literature because nobody else is doing this kind of quantifying,
Hmm.
and
Hmm.
they're not also not asking how much time there was or how much bioturbation because that's they
Yeah.
already know. And a person believer in the standard model already knows how many millions of years it was. And so, and they're not going to ask whether that's correct or not because they've already are committed to that. So they're not doing the kind of work that we're doing because it would make no sense to do what we're doing if you already firmly know that it was hundreds of millions of years. And
Mm.
so, and I'll put in another aspect here that's important at this
Mm.
point. So what was our ultimate reason for doing this? Well,
Mm.
of course, the standard measure of time in the in geology is the radiometric dating scale, and this is one of the big items that convinces a lot of people that their Bible story of a short time can't really be true. And this is one of our we wanted to tackle that. What can we say about that? And there's radiometric dating is a very well known and work. But there are aspects of it that really people don't think about or don't realize. There's no way you can take a rock and put it in any instrument that you have or you could even invent and tell you how many years old this rock is. Rocks don't have that kind of information in them. You measure ratios of daughter product and parent element, and those are the ratios. That's what you get is ratios, and then you make some assumptions and calculate an age. Okay, and that's one that's one of the limits of this method. You cannot it cannot determine actually you cannot quantify an age. You have to calculate that based on your assumptions. And the other thing about it is that for this to be a really reliable method, you have to be able to somehow test this idea. Okay, how are we going to go back in time and measure how much actual time has passed? We can't do that. We weren't there. We can't do that. You cannot test the actual accuracy and reliability of radiometric data. It's not possible. Even though we would like to, but it's assumed to be true. And so it'd be good to have other methods of measuring time that we can compare that don't have the same difficulties as radiometric data. And this is one of those, and that's why we did it. It gives you
Uh-huh.
a measure, some estimate of time that is not dependent on the assumptions of radiometric data. And so that's
Yeah.
why we do it.
Just to sort of underline for our viewers who've been following this, I want to come back to your findings there. So 97% of the formations that you studied, uh, the bioturbation intensity was level 1. So that means that there may have been some burrows present, but they weren't enough to disturb the sedimentary layering.
No, you might have
Uh,
had one or two burrows in your photograph that you took, but that's it.
yeah.
So when you
And
say 97%, are you talking about with respect to the thickness or with respect to the
That's
number of things?
that's a measure of thickness. Yes,
Wow.
97% of the
Oh.
of
wow.
the
Oh, okay. So
yeah,
it's
level one.
Yeah.
it's actual thickness of sediment, not just presence or absence in a particular rock layer. Wow.
Wow.
Okay.
And less than 1% of the thickness was completely homogenized, was level 4. I mean, that's, to me, that's astonishing. Given, what I read in that textbook by Bromley, that we would expect, given, modern processes, that 100% of sediment is going to be intensely bioturbated. And you're telling me that, your discoveries indicate that actually there's very low intensity of bioturbation through most of the record that you studied.
That's right.
Wow.
I mean, this is just mind blowing
It,
to
it,
to
it
me
is
as a
mind.
geologist.
Yeah, and it's mind blowing to me just as an observer because as you mentioned, Paul, the anti-creationists come in and say, "Oh, you can't explain bioturbation, and bioturbation is everywhere, and it's ubiquitous, and it's in all these layers and all this stuff." And you get sort of, for those of us who aren't very, you know, familiar with these kinds of things, it sounds intimidating. It sounds like, oh well,
Mm. Mm.
I guess things must be wrong. I guess there must be a lot of time recorded in the fossil record. But when you actually go out and measure it, that does not seem to be the case.
Yeah.
And if we can ask, okay, have we missed something? Is there any evidence that could verify that this is realistic? And as a matter of fact, if there was a lot of bioturbation, it should have destroyed the layering and the setup, the divisions between layers and stuff. Well, you look at these formations one after another, and you just don't see that.
Mm-hmm.
The 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 rocks have very clear layering in them, and this is this is what you find normal. That's very clear, and so, and we look at these formations that we measured, and in our publication, we have pictures of representative rock formations, and the layers are all there. It's just
Mm.
very clear, and that verifies. That's a strong verification that our data are reasonable, are right.
Yeah.
It just the bioturbation isn't there.
Yeah. Yeah.
It didn't destroy these layers. The layer.
So is there?
And
Do you think?
sedimentary geologists are very happy about that, because if the layering was all destroyed in the sediments,
right.
it would be very hard for them to do their job, because sedimentologists rely on studying the sedimentary structures to sort of understand what the processes were that deposited the sediment, right?
In normal conditions, all of those structures should have been destroyed by bioturbation.
Mm-hmm.
And so geologists, sedimentologists don't realize that they are able to do their work because of the flood, because that kept all these rocks from being bioturbated, their sedimentary structures from being destroyed.
Wow.
But yet that's the way it is.
Now, could there be, so I'm going to play devil's advocate just for a second here.
Sure.
Could there be some kind of preservation bias against bioturbated sediments? Is it possible that somehow bioturbation just makes a sediment not preservable? I guess I don't know how to say that, but not preservable. Is that a reasonable guess?
I think not, because the more bioturbated it is, you might be even easier to destroy it. Anyway, I don't know of any reason why there should be a connection like
Yeah. Okay.
that. The big question is, why have other people not seen this low level of bioturbation?
Because they're not
And I think
looking for it.
yes, I think there is a definite reason. One of the papers I have on bioturbation is discussing why there is such a low level of bioturbation in the Paleozoic. And he comments that paleontologists are normally not going to spend time doing research on formations that have um, no or very few fossils, which would include bioturbation.
Yeah.
So that would be unproductive research. So they're not going to bother doing that. And so I think there is a natural bias against people doing research on these formations that don't have much. If a formation has a few levels where there's plenty of bioturbation, that's where they'll do their study. You wouldn't waste your time studying all this rock with no bioturbation. That would be unproductive. And so there is a natural, I think, research bias against noticing these non-bioturbated rocks, unless you have a reason, like believing in the Bible, a
Yeah.
reason for thinking it's important.
Makes sense.
And in terms of the flood, um, how do we explain those occasional levels where there is more intense bioturbation? How does that kind of fit into your flood perspective?
I see that the processes during the flood, as I understand them, are not like a continuous thing. There'll
Hmm.
be some little breaks here and there where something words, and those will be the times you can have a few centimeters of bioturbation.
Hmm.
Um, but
Yeah.
it's not going to be continuous.
Hmm.
Yeah.
Yeah.
I think we have an awful lot to learn about what's really happening during the flood. It's a
Hmm.
complex event,
Hmm.
and you
Yeah.
can have things happening.
Yeah. Yeah.
Yeah. Yeah. I think we do have that sense of at least that's the sort of when a skeptic is talking to me about the flood, it usually is that simple. Everything gets jumbled up in the water at one time and then gets dumped at one time. And I just feel like, how is that realistic at all? It's a year-long event here, and there's regression and progression of the water, and I just don't see how scrambling everything up at once and dumping it in a single place is the right way to think about things.
Yeah,
Yeah.
well, people who will say that probably have not really tried to truly understand what the flood could have been like.
Mm.
That they're just think it's nonsense, and yeah, well, it must be just chaos.
Mhm.
Wasn't chaos.
So, Leonard, you said that your particular sort of project has come to an end. You've done what you're going to do on this topic. Um, do you have plans to sort of publish this work, and uh, do others have plans to maybe follow up with further research? What kinds of questions could they potentially explore to build on what you've done?
Yeah, well, okay. Dr. Chadwick and I have puzzled a little bit and argued a little bit about whether we should try to publish this in a standard research journal. I think they'd never look at it. He thinks it might be worth at least trying, so at least get the reviewers aware of this. But we didn't bother to do that. We have published this in a new creationist journal
Uh-huh.
called New Creation Studies, um, and so it is published and available.
And what questions do you think remain open that maybe students could take up and build on the work that you've already done?
Well, I mean, we did all these formations in one part of the country. There's all the rest of the world to study. I mean, I don't know how well things are preserved in other parts of the world, um, but one question might could be of interest. Okay, we studied up through the Eocene, and there's just a couple of formations in the Eocene. Okay, that's in the part of the record where we like to figure out where the flood ended.
Mm-hmm.
And so I think an intensive study of this type, just in the Cenozoic, the upper part of the record, might be productive to understand, to help us to understand what parts of the record are in the flood and what are in the transition times after and what after the flood.
Hmm.
There, I would expect there to be you start finding more bioturbation up there
Yeah.
after the flood. So that I think
Yeah,
would one productive project.
that makes sense.
Yeah,
Yeah.
that sounds like a hot topic. It seems like we've done that before here on the podcast, talking about where the flood ends in the geologic record.
Yeah, yeah. That's brilliant. Todd, any last questions before we begin to sort of wind this up?
Well, I think you know, I'm just sort of trying to wrap my head around this because it's such a big deal. I have
Mm.
for years I've been, going around on geology field trips with geologists pointing stuff out to me, and I know nothing. I don't know sandstone from limestone from anything. I know nothing. So the one thing that I have noticed is that there's usually these just absolutely massive sections, vertical faces of rock, and yeah, there's either this fine layering or it's just this one big thing with basically nothing in it, leaving me to wonder maybe I'm just not seeing it. Maybe, a real geologist would come along and look at this and they would see, oh, here, look at over here. Here's a burrow that you didn't notice because you don't know what you're looking for. That sort of thing. And I think that kind of healthy skepticism is important because I'm not a geologist, right? So,
Mm.
but now you're telling me that this sort of intuitive perspective on the fossil record seems to be statistically correct, that there is just a huge lack of bioturbation that's just massively noticeable and
Mhm.
unmistakable and seems to be the majority pattern of the fossil record is not actually fossils.
Unmistakable if you're looking,
Yes,
asking the
if you're
right
looking
question.
for it. Yeah, yeah.
Yeah.
It
Yeah.
feels like we should do this
Yeah.
with body fossils too. Because I'm
Well,
wondering,
if
do body fossils come in specific high density horizons? I know we have places where we go to find body fossils. We kind of know here's this place that has a lot of dinosaurs, and here's this place over here that has a lot of something else, mammals or whatever. Are they going to be similarly distributed in these high density deposits, or are they uniform throughout the fossil record? I think that sounds like an interesting corollary question too. Do you know if anybody?
probably harder to study. And one difference is to keep in
Yeah.
mind: a body fossil means you had an animal there.
Mm-hmm.
Bioturbation, you have to have live things moving to get
Mm-hmm.
bioturbation.
Mm-hmm.
So that's a difference.
Yeah, that would definitely be a difference. It would definitely be a difference. But
Yeah.
it seems to me that, just knowing the density, the general density of fossils over time in the fossil record would be a valuable thing to know.
Yeah.
Yeah.
Just
Yeah.
generally speaking. I don't know. I'm a biochemist. What do I know about
Oh,
fossils?
that's smart. Biochemists can figure out things that I can't.
Yeah. Yeah, well,
That
sure.
one question that would puzzle us to, well, we've talked about how we should say this. People will ask, have you proven the flood?
Yeah.
Well, the word proof should not be used in science.
Right.
Right.
And certainly not, no, I think our evidence is very strongly pointing towards rapid processes, very rapid processes, very widespread. Whether you're going to put that in the flood, we can't really demonstrate that, but we can certainly say our data are incompatible with the radiometric time scale.
Mm-hmm. Mm-hmm. Yeah. Yeah, people always want to know if we've proved the flood, and we always try to remind them on this show. It's very hard to imagine a single study that would definitively prove the flood. Maybe if we found Noah's Ark with fossilized Noah's family in it, but I doubt that's going to happen. Even then, you couldn't prove a global flood. You just proved there was somebody in an ark. So, it's very hard to imagine one thing that would just definitively clinch it for us.
But I do want to sort of underline to our viewers and listeners how surprising the results of this study really are.
Yes.
If you read the literature and I'm sure you're right, Leonard, that there's this kind of bias because people if they're wanting to study trace fossils, they're going to go to layers which have bioturbation in them, have trace fossils.
Yeah.
And so they're the ones that you get all the lovely photographs, in the textbooks. But people haven't noticed that actually these are very unrepresentative based on the kind of quantitative survey that you've done. And it would be great to see that replicated, maybe in some other places, to see whether that holds up. Yeah.
Yeah, that's another good idea.
Person I know that took a class once in trace fossils from a very prominent researcher in that field. And this person just made a comment one day in class. So they said, I don't lose sleep over it, but I wonder why there isn't more bioturbation out there.
Yeah.
Hmm.
Yeah. Yeah. Good question. Yeah, that's really good. That's fascinating. I mean, that's been brilliant. And of course, you're right to sort of remind us at the end there that this is not the silver bullet that kind of proves the flood.
Right.
But it is a challenge. It's a significant challenge to the radiometric timescale. And we've been doing a series on the podcast about the radiometric timescale. And this is another one of those pieces of evidence, rather like the ones that we talked about with Monty Fleming, Todd, you'll remember.
Yeah, exactly.
Where we have geological processes that present a challenge to the kinds of ages that you get from the radiometric
Right. Exactly.
methods.
Yeah.
So, yeah. As we close out, I just wanted to recommend to our viewers and listeners a couple of things. I'm sure Leonard won't mind me recommending his textbook, Faith, Reason and Earth History. This is the third edition, I think, and it's available as a free ebook. You can get the hard copy like this, but it's also available online. We'll put the details of that in the show notes, but this is highly recommended. And it does have a section in here about the bioturbation work that Leonard has been talking about. And I also, now I must confess, I haven't read this yet, but I recently got a copy of this book, Rising Waters, Enduring Faith. And Leonard is one of the contributors to this edited volume. And again, he does cover the bioturbation argument in one of his chapters in this book. And browsing through the contents page, this looks like a really worthwhile read. So do look up those books if you want to find out more about this topic.
If you don't mind, I'll mention there's a
Sure.
book. There's a book in production right now that is, well, the title is Geologic History: Insights from Genesis and Geology. It's a report of the 50 years of research that Chadwick and I have done,
Wow.
and it's being produced actually by Thomas Purifoy and his group.
Oh, great.
So, and I think it'll come out sometime this summer.
Fantastic.
That sounds great. We'll look forward to that, certainly. Thank you so much, Leonard, for being on the podcast today. It's been a long time getting to this point where we could have you as a guest, but I'm so glad we have. And I hope maybe at some point you can come back on and talk to us about some of the other research projects that you've done, because they're all just as interesting as this one. So that's great. Thank you so much. As ever, we don't know what's coming up next on the show. We don't know what's next on the schedule, but I'm sure it will be interesting. So join us then. We'll be back in a couple of weeks' time, and we'll see you then.
See you later.
Thanks for listening to this episode of Let's Talk Creation. For more information, visit us at letstalkcreation.org, where you'll find an archive of past episodes and all our show notes. If you'd like to leave a comment or make a suggestion, you can find us on all the major social media platforms. Let's Talk Creation is brought to you in the U.S. by Core Academy of Science and in the U.K. by Biblical Creation Trust. As a listener-supported ministry, we are grateful for all of your financial support. Find out how you can make a contribution at our website, letstalkcreation.org. Also remember to like, subscribe, and share this episode with your friends. Thanks, and see you next time.